Good day. I'm very excited to deliver this lecture, and I'm going to talk a little bit about how we were able to accelerate COVID vaccine development because of the lessons we've learned from HIV. As a background to this lecture, I want to first emphasize that the COVID-19 pandemic has devastated communities and countries across the globe, and that this pandemic has also impacted on health service delivery, especially in low and middle income country settings, that the decades of investment in HIV research has supported COVID-19 research, and that the rapid establishment of efficacious COVID-19 vaccines establishes a proof of principle that can be deployed for when we have an effective HIV vaccine. I'm going to give a bit of background about the impacts of COVID-19 in South Africa, especially in terms of health delivery and HIV, and poverty that has ensued because of lockdown issues are related to trying to contain the pandemic in South Africa. So first of all, we see right in the beginning, as we go into lockdown in South Africa, a huge hesitancy to use care. And we can see that in HIV-infected women, there was a decline of use of ART during pregnancy, one because people were scared of getting coronavirus. Sometimes antiretroviral therapy was not available. People didn't want to wait in health systems. There were transport issues, and people also, we noticed that pregnant women weren't accessing antenatal care very well during lockdown. And this was because mostly they were afraid of getting coronavirus and had transport issues. If we look at the impact of COVID-19 on HIV, we definitely in South Africa, right in the beginning, saw a breakdown in the number of people accessing viral load testing and the number of people getting CD4 tests. So this slide demonstrates how during level 5 lockdown, there was a huge decline in people getting CD4 tests and HIV viral loads, which impacted on care and hopefully did not translate into not getting antiretroviral therapy. But we'll only know the brunt of the impact of the pandemic on HIV at a much later stage as we start to reassemble our health system. We also saw huge poverty indicators and saw an increased amount of people in South Africa running out of money to buy food, going hungry in the last 7 days, and children in the households going hungry. And so not only did the pandemic affect chronic diseases like HIV and TB, but also had an impact on household food security. So now I'm going to move on to talk about how HIV research assisted COVID-19 vaccine development. So HIV research is the gift that keeps on giving. Because of the investment in HIV research, both in basic science and laboratory capacity, in HIV vaccine development, in HIV clinical trials platform expertise, the statistical expertise of many people, particularly in SHARP, and the widespread community engagement created a necessary and important platform with which we could move ahead with COVID vaccine research. So in terms of COVID-19 vaccine development acceleration, the decades of investment in the HIV response did create platforms that supported COVID-19 responses. The HIV research infrastructure was leveraged to accelerate COVID-19 responses, and the HIV lessons that we learned influenced the responses to be human rights focused and address the issues of equity. In terms of HIV research, we were able to use the novel vaccine platforms, design of clinical trials, and our molecular surveillance to support the work done in COVID-19. The ineffectiveness of the GP120 and GP160 HIV vaccines caused the expansion of the vaccine platform development to expand to DNA and mRNA vaccines to utilize vector-based platforms like the adeno5, the A26, and chimp adeno, and also the beginning of an evaluation of monoclonal antibodies to prevent HIV. And all of these strategies helped the clinical development of vaccines and monoclonal antibodies. But also in terms of SARS-CoV-2 vaccine design, the lessons learned from structure-based designs on stabilized epitope scaffold proteins were utilized, and this was based on experience in HIV. And then again, the global molecular surveillance developed for HIV was utilized, and this is how we were able to evaluate the emergence of variants of concern, particularly the variant of concern in South Africa, the B1351 or the 501V2 mutation, now known as beta variant of concern. And this was very important because the very basis of the surveillance in South Africa, because of HIV, allowed us to do molecular surveillance and able to identify variants of concern very, very early on in our epidemic towards the end of last year. If we look at how COVID vaccines were accelerated, this slide shows a science article published by Larry Corey that looked at developing a pathway for an effective vaccine for SARS-CoV-2, but also understood that we required both industry, government, and academia to collaborate in unprecedented ways, each adding to the individual strengths. And so a very important platform was created that we were able to rapidly evaluate vaccine trials and to accelerate the licensure and the distribution of these vaccines. And this kind of approach that goes beyond just clinical trial vaccine trial development, but goes towards emergency use authorization, and the rollout will bode very well for an effective HIV vaccine. So in terms of the conceptual framework for COVID-19 vaccine development, the most important concept was that the need to develop multiple vaccine platforms simultaneously. So unlike HIV vaccine research, where we've gone from one vaccine platform to the other, this approach was to deploy multiple vaccine platforms to find a solution as quickly as possible. And it was important because we had to utilize multiple vaccine platforms because we would have to be able to manufacture lots and lots of vaccines to support the use of vaccines at a global level, to make sure that everyone on the planet could get vaccines. We also had to make sure that the platforms we knew them for safety, as well as scientifically. And we also had to make sure that whatever was tested, we could scale in terms of manufacturing. And so it's very important to know the safety and to make sure that we would be able to scale up its production. We also knew that you needed a coordinated approach, and the US government utilized this approach to make sure there was global vaccine manufacturing companies that were being used. And this was an unprecedented coordinated approach that was used to test, manufacture the vaccine at scale, and to try and deliver vaccine into people's arms throughout the world. And more or less in the high-income countries, this has been achieved, but we have not seen this translate into many especially in Africa, in our continent, which I will show you later. So in terms of the goals of Operation Warp Speed, so I'm going to use this as a great example. The various technology platforms that were utilized were looking at protein vaccines, and some of them went sped ahead. And we know vaccines like Novavax have shown efficacy already, and that some other vaccines haven't been been able to get into the field as quickly as possible. And we know that one of them was the Sanofi vaccine. But the viral vector platforms, the Ad26 and the ChIMP-Ad vaccine approach, got into clinical trials quite rapidly. And by the end of last year and early this year, we knew the results of these studies. And then obviously the most important expansion and the most promising technology for the future has been the RNA platform and technology development that have enabled the development of the mRNA Pfizer and Moderna vaccines. So in terms of trying to organize this approach, it was very important to harmonize efficacy trials. So to take the RNA, the ChAdOx, the Ad26, the nanoparticle, and the prefusion spike recombinant protein platform, one had to look at how you harmonize these efficacy trials to try and compare. And it was important to have clinical trial networks that were operating together to make sure that there was harmonized endpoint data so you could compare from trial to trial, to make sure that common labs were used so that you could define the infection, that you could look at the responses to the spike protein and the spike epitopes, and look at T-cell response in labs that were comparable. It was very important for this because we also had to develop correlates of protection across protocols, and particularly moving forward when you look at immunobridging. And the most important thing for all of this was to have a common DSMB that could evaluate and keep an eye on all the trials as the efficacy data was emerging. And so very important to have a harmonized approach to rapidly develop various platforms in COVID vaccine research. So it's very important to balance the clinical trial design. And the main goal was because there was going to be multiple vaccine platforms being evaluated. And so roughly each trial was designed to enroll at least 30,000 people, to have 150 endpoints, and also to make sure that, that we enrolled people that were at risk for severe people with comorbidities and groups that were at risk for COVID disease. So it is important to make sure that they were enrolled. It's also important to expand, not just in the US, but to expand to other populations in Latin America and Africa to understand population diversities and variants of concern. And it's also important to evaluate vaccines in the epidemiological setting of people at risk of its complications, including comorbidities, age, and race. So what happened was the whole HIV vaccine platform, HIV Vaccine Research and HPTN, worked together with other consortiums, including the AIDSImpact and the Department of Defense, to create a COVID-19 prevention network at a global level. And this slide just shows the extent of the COVID-19 prevention network that was funded by the NIH, and shows sites both in North America, South America, and Africa. And this was very important to develop capacity at a global level to understand the vaccine platforms in the various areas and the circulating variants of concern. This just shows the slide of the expansion in sub-Saharan Africa, and wherever there was an HIV vaccine clinical trials site, we were able to expand our CoVPN and COVID-19 vaccine capabilities and vaccine research. So there was a huge investment that has been made in the COVID-19 vaccine pipeline. In the beginning, around 280,000 participants, with 33 vaccine candidates in 470 sites in 34 countries, worked together to try and answer the question about the vaccine platform and to try and find an answer in less than a year. This huge collaboration to find a vaccine was very important, but we did find that South America and Africa were behind the pack, and that in the end, the 7 candidates that were supported by the Operation Warp Speed, aiming to deliver 300 million COVID-19 vaccines with a US dollar bill of 10 billion rand, $10 billion was executed. But you can see from this slide is that South Africa and Africa were underrepresented, as well as South America. And this is a good indication for future pandemic responses to make sure that critical continents capacity is developed as well. This slide just describes the rapid acceleration of the mRNA platform and shows the efficacy of both the Pfizer mRNA 1273 vaccine and the Moderna one, and shows the success of mRNA in reducing reducing COVID-19 infection in these cohorts, which set the pace for emergency use authorization and its use and deployment in high middle income countries, starting from December last year. This year, we also saw the efficacy of the AD26 COVID-2 vaccine, and this was a vaccine trial that was conducted in 3 continents, North America, South America, and Africa, and enrolled over around 45,000 individuals. And to answer a question about the efficacy of this vaccine in areas of the world that had other variants, like in Brazil and South Africa. The important thing about this vaccine is that the AID26 platform has been used in HIV vaccine research, and we are busy still rolling out evaluating the AID26 HIV vaccine platform in efficacy studies both in sub-Saharan Africa and in the Americas. So how did all this translate into implementation science? So the expertise that we saw in HIV research were leveraged because a lot of the HIV researchers had gone from clinical trial research to operational research when we had to roll out mother-to-child interventions and antiretroviral scale-up in our country. And so we used this very same platform to roll out the first phase of COVID-19 vaccines in South Africa. And we started a program called the Sosonke study. And this basically is an implementation study that involved 500,000 healthcare workers. And we were able to deliver half the Sosonke AID26 single jab vaccine to healthcare workers from about the 17th of February until the 14th of May. And this established the platform for vaccine rollout in South Africa. This slide demonstrates the various approaches that South Africa will use in its deployment of COVID-19 vaccines. And the Sosonke implementation study was started in mid-February and served to roll out the AID26 vaccine to healthcare workers in South Africa. This is the first time we've been involved in such a huge implementation study, and we used all the expertise of our HIV vaccine researchers. So we had to scale up our pharmacies in parts of the country where there was no research capability. Because this was an implementation study, we had to have research jurisdiction. All the pharmacy staff had to have GCP trained and be protocol trained, and we used the Sosonke pharmacy work packages and training materials to roll out even to urban settings. And this just shows you, the slide shows you how we had to make makeshift pharmacies in rural parts of South Africa. This slide shows the kind of steps we had to use for the implementation science Sosonke study. And the first section of the slide shows the healthcare workers coming to enroll into the Sosonke study, and the group to informed consent, and the waiting for the enrollment and the validation of the informed consent. We had to create a vaccination room or a section of a hall to make sure that we could deliver the vaccine program. And then we also had to create an observation area with an emergency section should there be any side effects. And this slide just shows you the implementation of the various sectors of the program to ensure that we could deliver the vaccine effectively and safely. We had to collect the vaccine. This slide just shows the scientists collecting the vaccine at the airport, and also that we had to fly the vaccine across the country to the more rural parts of South Africa to ensure that there was equity in our execution of this implementation study. This slide shows all the participants. These are healthcare workers coming from all parts of the provinces, coming in buses, queuing and waiting to be vaccinated, and just shows you that you're able to do implementation studies even in rural parts of the country. These are some pictures of the process to get vaccinated in various parts of our settings, and most of the times we use big halls that we're able to where we could implement social distancing and make sure there was good and adequate ventilation. This just shows you some of the targets. Every day we set targets for vaccines, the number of people that we targeted, the number of vaccines, and the number of vaccines in the fridge. And this just shows you the kind of teamwork you need when you do implementation science. These are healthcare workers waiting in a hall in Cape Town to be vaccinated in the screens just behind this picture. So this shows you an example of all the implementation work that we've done in South Africa, but this slide shows you how underrepresented Africa is when it comes to COVID vaccine rollout, and that there's a lot of work to be done to translate the successes that we've seen in the last year into global spread and distribution, and how some countries lag behind and have not managed to secure enough vaccines for their populace. So in conclusion, I want to say that HIV vaccine research has enabled the progress made to establish the efficacy of COVID-19 vaccines, and that the rapidity of COVID vaccine development and distribution has shown us that the urgency to end an epidemic can be successful, and that funding for research can be found within months, and is a good example for both HIV and TB vaccine development, that where there's a will, there's a way. It also has shown us that the development of the vaccine has proved that public institutions, universities, pharmaceutical companies, and non-profit organizations can all work together to create lifelong technologies that can hopefully translate into HIV vaccine research and TB research. But what we have seen is that low middle income country settings have lagged behind in access, and this needs to be addressed for future research and future pandemics. That vaccine, we've seen also that vaccine production in Africa has become a hot topic, and this has been emphasized because of the persistent struggle for most African countries to secure COVID-19 vaccines. Hopefully, HIV vaccine research and vaccine development must learn from the rapid process of registration and implementation of adult-based vaccine programs for the future, and that what we see is a nice cycle, that how HIV vaccine research helped COVID vaccines, and with the emergency use authorization and the deployment of adult-based vaccine programs has given us a platform to move forward when we have an effective HIV vaccine in the future. I'd like to thank many people, particularly the HVTN and the CoVPN, DAIDS, for doing a lot of the funding, the Vaccine Research Center that were critical for developing the immunogens and the platforms, obviously NIH with Tony Fauci and Francis Collins, Operation Warp Speed in South Africa. I'd like to acknowledge Linda Gale Becker, Nigel Garrett, Amina Gogo, Ian Sana, and the Sasonki study team, as well as the ensemble study team that were imperative for our expansion of the AID26 platform in South Africa. Thank you very much.